Okay, let's just dive right into this explainer by confronting a massive biological impossibility. If you were to somehow take all of the DNA molecules in a single microscopic human cell, completely unwind them and place them end to end, they would stretch to about 6 feet long. Just take a second to visualize that. Literally 6 feet of genetic material, which is taller than a lot of us, crammed into a single, invisible cell.
So that wild statistic brings us to the central mystery we're tackling today. How in the world does the cell manage to securely package such an enormous amount of genetic information into a microscopic space? And we aren't just talking about stuffing it into a tiny box. This DNA has to be perfectly organized, highly protected, and most importantly, readily accessible for the cell to actually function.
Blueprint of the nucleus, inside the biological vault. To figure this out, we're shrinking down to take an architectural tour of the cell's ultimate secure command center.
Now, first things first, we have to establish that this highly secure vault really only exists in a specific type of cell, a eukaryotic cell. Unlike simpler organisms, eukaryotes possess a well-defined, membrane-bound nucleus. It acts as a dedicated fortress, specifically designed to house and protect the genome, which for eukaryotes is made up of double-stranded linear DNA molecules.
It's a brilliant contrast in cellular architecture. While prokaryotes, like bacteria, have an open floor plan where their single circular DNA molecule is supercoiled in a region called the nucleoid, eukaryotes lock theirs away. Prokaryotes just twist that DNA coil on itself like a tight rubber band to save space. Eukaryotes, however, evolved a much more complex, enclosed system for their massive amounts of linear DNA.
Section 1. Breaching the perimeter. The nuclear envelope.
Our first stop on the tour is the fortified outer wall of this command center. You obviously can't have highly sensitive blueprints exposed to the bustling factory floor of the cytoplasm, so this perimeter is serious business. It's protected by a parallel double membrane system. You have an outer membrane, which is actually continuous with the endoplasmic reticulum and even has ribosomes on it, and an inner membrane. Between them is a tiny gap measuring just 10 to 50 nanometers, known as the perinuclear space. This whole setup forms a powerful barrier.
But you still need things to get in and out, right? Enter the specialized security gates, the nuclear pores. At various points, the inner and outer membranes fuse together to create these minute interruptions. But they aren't just open holes, they're strictly regulated passages. They control the two-way traffic of crucial molecules, specifically RNA and proteins, moving back and forth.
Section 2. Inside the command room. Navigating the nucleoplasm.
Once we slip past those heavily guarded nuclear pores, we're inside the command room itself, which is filled with a clear fluid matrix called the nucleoplasm, or nuclear sap. Floating in here, we find the nucleolus. Now, unlike the nucleus, the nucleolus isn't membrane-bound. It's a highly active factory area right in the middle of the room. Its primary job is active ribosomal RNA, or rRNA, synthesis. Actually, cells that make a lot of protein will have larger and more numerous nucleoli. The nucleoplasm also holds a network of protein fibrils called the nuclear matrix, which helps maintain the shape of the entire nucleus.
Section 3. The ultimate secret. Solving the DNA packaging problem. All right, here's the main event. Discovering exactly how the cell solves the engineering nightmare of packing those 6 feet of linear DNA into this tiny room. The secret is an elegant 4-step packaging process that systematically compresses the DNA down to a fraction of its size. Eukaryotic DNA is simply too long to exist without a dedicated strategy, so it relies on a structural hierarchy that folds and coils the DNA tighter and tighter.
Let's look at how this builds from the ground up. DNA is an acidic molecule and carries a negative charge, thanks to its phosphate groups. To pack it, the cell uses proteins called histones, which are basic and carry a positive charge. It's a perfect molecular attraction. The negative DNA tightly coils right around the positive histone proteins. Think of this core as a microscopic spool that carefully organizes an incredibly long thread. Specifically, it's called a histone octamer. It consists of 8 histone molecules, 2 each of H2A, H2B, H3, and H4. The DNA wraps securely around this core to form a neat spherical structure.
In the early 1940s, when scientists first looked at this structure under an electron microscope, they saw something amazing. They described it perfectly as looking just like beads on a string. Fun fact, at the time, they actually thought each bead might be a single gene.
Today, we know each one of those beads is actually called a nucleosome. A single nucleosome involves about 200 base pairs of DNA wrapping around that histone octamer core, and the string between the beads is a small segment called linker DNA. To keep it from unraveling, there's an extra histone called H1, which isn't part of the core but binds to the linker DNA to lock the whole wrapped structure securely into place.
Section 4. Maximum compression, from chromatin to chromosomes. During a cell's normal resting phase, interphase, this packed genetic material exists as long, elaborate threads called chromatin fibers. This keeps the DNA packed away, but still somewhat accessible for the cell to use. But when the cell is ready to divide, that chromatin undergoes an extreme compression sequence. Those chains coil up even further into tight helical fibers and supercoils, finally condensing into the microscopically visible, slender structures we know as chromosomes. This is maximum compression.
These densely packed chromosomes are architectural marvels. When they appear during cell division, they essentially feature two identical chromatids held tightly together at a primary constriction, or pinch point, called the centromere. On the sides of this centromere are disc-shaped structures called kinetochores, and the exact position of that centromere actually forms the basis for how scientists classify different chromosomes.
So, having explored this ultimate biological vault, from the double membrane perimeter to the intricate spool-like nucleosomes compressing 6 feet of DNA, we have to leave you with one final, mind-bending thought. If the DNA is wrapped up this tightly around these histone proteins to save space, how does the cellular machinery ever manage to pry it apart, read the instructions, and actually express its genes? That dynamic tension between extreme security and constant accessibility, that is the true genius of cellular engineering.
Thanks for watching, friends. Don't forget to like and subscribe. See you in the next one.